bacterial cell membrane. Bacterial cell membrane, it regulates what moves in and out of the cytoplasm. Plasma membrane is a living membrane. Bacterial cell membrane have the same general structure as the membrane of all other cells. It consists of a lipid bilayer with numerous proteins dispersed in it. Bacterial cell membranes are composed of about 40% phospholipid and 60% protein. One of the most dynamic structure in prokaryotic cell, the plasma membrane mainly functions as the selective permeable barrier that regulate the passage of substances into and out of the cell wall. The bacterial membrane allow passage of water and uncharged molecules up to molecular weight about 100 Dalton. It does not allow the passage of larger molecules or any charged substances except by means of special membrane transport process and transport system. Since prokaryotes lack membrane bound intracellular organelles for the process of respiration or photosynthesis or secretion, so the plasma membrane performs this process for the cell. Because, for example, at the time of aerobic respiration, during the electron transport system, ATP synthesis is found in the prokaryotic cell membrane. The photosynthetic chromophores. These chromophores are present in certain bacterial species. These are act as the similar function to chlorophyll found in plants. These photosynthetic chromophores in some bacterial species during the electron transport chain system, during the aerobic respiration time, the ATP synthesis, the chromophores that harvest light energy for conversion into chemical energy are located in the membrane. Hence, the plasma membrane is the site of oxidative phosphorylation and photophosphorylation in prokaryotes. And similar, analogous to the functions of mitochondria and chloroplast in eukaryotic cells. So, similar to eukaryotic cells, the bacterial plasma membrane function similar to the eukaryotic mitochondria and the chloroplast. ഇവിടെ ബാക്ടീരിയയുടെ പ്ലാസ്മ മെംബ്രെയിൻ യൂക്കാരിയോട്സിൻ്റെ മൈറ്റോകോൺട്രിയയും ആനിമൽസിലുള്ള മൈറ്റോകോൺട്രിയ ചെയ്യുന്ന ഫംഗ്ഷനും പ്ലാന്റ്സിലുള്ള ക്ലോറോപ്ലാസ്റ്റ് ചെയ്യുന്ന ഫംഗ്ഷനും ചില ബാക്ടീരിയൽ സ്പീഷീസിലെ, അതായത് ഫോട്ടോസിന്തറ്റിക് ക്രോമോഫോഴ്സ് ഉള്ള ബാക്ടീരിയൽ സ്പീഷീസ് ചെയ്യും. അപ്പോൾ ഇങ്ങനെ പ്രൊഡ്യൂസ് ചെയ്ത ATPയെ പ്ലാസ്മ മെംബ്രെയിനിലാണ് ഇതിനെ സൈറ്റോപ്ലാസത്തിലേക്ക് ട്രാൻസ്ഫർ ചെയ്യണം. സോ അതുകൊണ്ട് നമുക്ക് പറയാം, ചെറിയ ചെറിയ മോളിക്യൂൾസിലൊക്കെ ബാക്ടീരിയൽ സെൽസിൻ്റെ സസ്റ്റൈനബിലിറ്റിക്ക് ആവശ്യമായ മോളിക്യൂൾസിനെ പ്ലാസ്മ മെംബ്രെയിൻ ഈസി ആയിട്ട് കടത്തിവിടുന്നു. The other functions attributed to bacterial cell membrane are synthesis of cell wall components, assisting in DNA replication, secretion of protein, serving as anchors for flagella, and detecting and responding to signals in the environment by special membrane bound receptor molecules. So, what are the major functions of bacterial cell membrane? And the bacterial cell membrane have functions like their synthesis of cell wall components, assisting DNA replication, secretion of protein, serving as anchors for flagella, and detecting and responding to signals in the environment by special membrane-bound receptor molecules present in the plasma membrane. So, diffusion is the changing of materials inside the cytoplasm and outside. This is based on the concentration gradient. The time of diffusion, particles move in from high concentration to its lower concentration. And the next is the osmosis, is the diffusion of water across a semi-permeable membrane. On the basis of the diffusion and osmosis, cells are divided into three: isotonic, hypertonic, hypotonic. Solutions are divided into isotonic, hypertonic and hypotonic. And in bacterial cell membrane possess passive transport, because selective particles are entering inside the cytoplasm via cell membrane, and also it take part in active transport. Example is the transport of ATP, and also it helps in engulfment, or that is phagocytosis or pinocytosis. Any foreign particles are entering or attacking the bacteria, so this plasma cell membrane can take part in the process known as engulfment or phagocytosis. It triggers the phagocytosis. And it also take part in the specialized group of translocation. Translocation means changing of some particles from one location to another location. So these are the major important functions of bacterial cell membrane. Okay, these are the detailed on bacterial cell membrane. And the next is the bacterial internal membrane system. Membranous organelles are absent from the cytoplasm of prokaryotes. However, some bacteria possess internal membranous structures. A commonly observed structure is the mesosome. Mesosomes are large enfoldings of the cell membrane in the form of vesicles, tubules or lamellae. These are often found close to septa or cross walls in dividing bacteria, and sometimes attached to the bacterial chromosome. The major functions of the mesosomes are, although not fully understood, but it is helping or attributed to cell division. Some species of bacteria, that is purple bacteria and cyanobacteria, possess internal membrane foldings. These foldings helps the bacteria assisting photosynthesis, because these mesosomal structures containing some chromophores, photosynthetic chromophores. This helps in the bacteria for photosynthesis. So in internal membrane system, first is the mesosome, and the second one is the pili. The bacterial appendage is pili or fimbriae and flagella. Pili are short hair-like protein, function as an adherence or adherence, or stick to each other, or stick to a surface, or harder to wash away. It helps for adherence or stick on the surface. It is also known as sex pili because it is specialized sex pilus. It is used for bacteria conjugation. It is a conjugation pili. Sometimes it is known as the conjugation pili or sex pili. It is a long tube-like projections connecting the protoplasm of two mating bacteria. These are fewer in number, approximately 1 to 10%. and helps in the transferring genetic material during conjugation. And the next is the bacterial. How this flagella, how this pili helps in bacterial conjugation. Bacterial reproduction, there are mainly two types. One is the asexual reproduction and another one is the sexual reproduction or conjugation. Bacteria divided by binary fission by asexually involves the copying of the DNA and splitting one parent cell into two new daughter cells. And in conjugation or sexual reproduction, one bacteria is able to transfer its DNA into another by means of a pili or pilus. The figure showing the conjugation or sexual reproduction in bacteria and asexual reproduction by binary fission. And the another internal membrane is the internal membrane system and structure is the flagella. Flagella is used for bacteria for motility. Most of the half of the non-bacteria have motile. Most of the motile bacteria move by use of flagella. Each bacteria may have one or two or many flagella. Based on the position and number of flagella, the bacteria are again classified into four. In some species of bacteria possess only one flagella. So this type of bacteria is known as Monotrichus. And some possess more than one at one position. So this type is known as Amphitrichus. And some species possess two flagella on opposite sides. This is known as Lophotrichus. And some species possess many flagella arising around the body, around the cell, is known as Peritrichus. In bacteria, flagella are long appendages which rotate by means of a motor located just under the cytoplasmic membrane, and the detailed structure of flagella indicated. Flagellum is made up of a protein subunits called flagellin. Each flagellum has a prominent filament, a basal body that is embedded in the cell, and a short curved hook that links the filament to its basal body. The diameter of prokaryotic flagellum is about 20 nanometer. The flagellar filament is rotated by a motor apparatus in the plasma membrane, allowing the cell to swim in fluid environments. In contrast to eukaryotic flagellum, the prokaryotic flagellum is not covered by plasma membrane. And the detailed structure of flagellum is and also the basal part is containing two types of ring, that is S ring and M ring. It acts as a rotor. It helps in flagellar movement. And also in middle part containing P ring, it is helps in bushing. And also in the upper part of the P ring containing L ring. And also the outer part, it contains a hook, a filament or propeller, and inside the region also a C ring. This is the detailed structure of flagella. The expected question is write down the bacterial structure of a detailed structure of bacterial flagella, and based on the number of flagella, based on the number and position of flagella, bacteria are classified into 4. Or write down the classification of bacteria based on the number and position of flagella. This is the bacterial structure of bacterial flagella. And next is the internal structure, the cytoplasm and cytoplasmic structures. Within the cell membrane is a semi-fluid cytoplasm is seen, mostly comprised of water and dissolved substance such as salt, protein, enzyme, lipids, and all biochemical necessary for its life. And inside the cytoplasm, a freely found genetic material is seen. Inside the cytoplasm, a nucleoid is the area containing the bacterial chromosome, that is bacterial DNA. The genome or the genetic material carrying information required for the consistent or highly essential activities of the prokaryotic cell is organized as a double-stranded circular DNA molecule that is freely found in the cytoplasm. That area is known as the nucleoid. As it is circular and continuous, it forms an endless chromosome. It is known as it is not considered as the true nucleus because it is freely found in the cytoplasm. The region where the bacterial chromosome is located is called nucleoid, and this is the structure of figure showing the structure of nucleoid. And the next and most important structure, internal structure found in the bacteria are plasmid. Inside the bacterial cytoplasm, an extra chromosomal DNA, not part of genome, but it contains different genes, is known as the plasmids. It is majorly used in the transfer process, that is conjugation across sex pillars, change in the genetic character of the recipient. It is the major tool used in the genetic engineering process or bacterial or rDNA technology. The major characteristic feature of prokaryotic cell is the presence of extra chromosomal genetic material called plasmid. They too are circular DNA molecules carrying genes for several activities not coded by the genes in the genomic DNA. അപ്പോൾ genomic DNAയിലുള്ള ജീനുകളല്ലാതെ ബാക്ടീരിയയ്ക്ക് സെല്ലിന് ഫങ്ഷൻ ചെയ്യാൻ ആവശ്യമായ എന്തൊക്കെയോ കുറേ ജീനുകൾ അടങ്ങിയ ഡബിൾ ഷേപ്പിലുള്ള ഒരു സർക്കുലർ ക്രോമസോമാണ് പ്ലാസ്മിഡ്സ്. ഇറ്റ്സ് വെരി ഇമ്പോർട്ടൻറ്റ്. Genes for antibiotic resistance, fertility, etc. are maintained in the plasmids. So, the number of plasmids in each cell may vary from 1 to several hundred, with average being around 20. These plasmids are capable of replication, which is independent of the replication of the genomic chromosome. Plasmids can be exchanged between cell and thus form a vehicle for the transfer of genes. In biotechnological application, these plasmids play the role of vectors that transfer genes into prokaryotic cells. അപ്പോൾ ബയോടെക്നോളജിയിലെ മേജർ ടെക്നിക്കുകൾ RDNA ടെക്നോളജി, ജീൻ ട്രാൻസ്ഫർ ടെക്നിക്കൊക്കെ ഈസ് ബേസ്ഡ് ഓൺ ദ based on the function of plasmid. Okay, so plasmids act as the tool for genetic engineering, especially gene transfer technique. So These are the major detailed structure of bacterial cell. And the another major important structure inside the internal membrane are the ribosome. Inside the bacterial cytoplasm, ribosomes are present. It's a site for protein synthesis. Some antibiotics work by messing with the ribosome. നമ്മൾ നേരത്തെ പറഞ്ഞു ബാക്ടീരിയയെ നശിപ്പിക്കുന്നത്, ബാക്ടീരിയൽ ഇൻഫെക്ഷനെ, ബാക്ടീരിയൽ കോളനിയെ നമ്മൾ നശിപ്പിക്കുന്നത് അതിൻ്റെ പെപ്റ്റിഡോഗ്ലൈക്കൻ ലെയറിനെ അറ്റാക്ക് ചെയ്തിട്ടാണ്. എന്നാൽ ഇത് കൂടാതെ ബാക്ടീരിയൽ സെല്ലിനെ നശിപ്പിച്ച് കളയാനായിട്ട് സ്പെഷ്യലൈസ്ഡ് ആൻറ്റിബയോട്ടിക് ഉണ്ട്. സോ അങ്ങനെ ബാക്ടീരിയ നശിപ്പിക്കുന്ന അതിൽ പ്രധാന റോള് വഹിക്കുന്ന ഒരു പാർട്ടാണ്, ഒരു സ്ട്രക്ചറാണ് ബാക്ടീരിയൽ റൈബോസോം. അപ്പോൾ സം ആൻറ്റിബയോട്ടിക്സ് ക്യാൻ ബി, ചില ബാക്ടീരിയയെ നമുക്ക് റൈബോസോമിനെ അറ്റാക്ക് ചെയ്യുന്ന മരുന്ന് കൊടുത്താൽ, ആൻറ്റിബയോട്ടിക്സ് യൂസ് ചെയ്ത് നമുക്ക് ബാക്ടീരിയൽ ഇൻഫെക്ഷനെ തടയാൻ പറ്റും. എന്നാൽ ആ മരുന്ന് മനുഷ്യരുടെ റൈബോസോമിനെ അറ്റാക്ക് ചെയ്യുകയും ചെയ്യരുത്. അപ്പോൾ സം ആൻറ്റിബയോട്ടിക്സ് വർക്ക് ബൈ മെസ്സിങ് വിത്ത് ദ റൈബോസോം, slightly smaller than eukaryotic ribosome. So antibiotic can work on bacteria but not affect us. So this type of identifying the toxicity is known as a selective toxicity. അപ്പോൾ ബാക്ടീരിയയിൽ ചെന്ന് ചില ഓർഗനെ മാത്രം നശിപ്പിക്കുക. അപ്പോൾ അങ്ങനെ ഈ റൈബോസോമിനെ ഡിസ്ട്രപ്റ്റ് ചെയ്യുന്ന സമയത്ത് പിന്നെ പ്രോട്ടീൻ സിന്തസിസ് നടക്കത്തില്ല. അപ്പോൾ ബാക്ടീരിയൽ സെല്ലിന് ആവശ്യമായിട്ടുള്ള പ്രോട്ടീനുകളും ഒന്നും നടക്കുന്നില്ല. അങ്ങനെ ബാക്ടീരിയൽ കോളനി നശിച്ചു പോകും. And inside the bacterial cytoplasm, some other structures are present. This is known as the inclusions or granules of sugar, lipid storage area. This is storage. Inclusion bodies are present in some bacterial species. These are act as the storage area. And also endospores. Example is the Bacillus and Clostridium species. Inside the bacterial cell, endospores are present. It's the hardest of bacterial structures. It is difficult to kill with the heat or chemical. These bacterial endospores are destroyed by autoclave at 120 degrees Celsius. So this outer endospore can be penetrate and destroy the genome of bacteria. So another bacterial internal membrane structure is known as the inclusions, and other structure is the endospore. The figure showing in